As you can see in Canvas, we have the next entry is the workshop. You want to maybe take in some questions first, or?
Yeah, let's see if there are some
Yeah.
questions.
Questions of or shared experience if anyone has tested deconvolution before.
Hi, I have a question. How do I know if what I'm seeing after the convolution is actually an artifact or it's my sample?
Yeah, very good question. I think if the point spread function of your scope matches the point spread function you use in the deconvolution, it's typically not a problem. And problems typically only arise if you try to push it too far. So here the different methods behave in different ways, but I would say all of them are iterative. So it does several iterations, try to improve gradually the output. And if you try to push them too much, to take too many iterations, you will start to see artifacts. And in most of the cases, it is that it starts to look noisy. So there will be super bright pixels here and there. So I think in general, you shouldn't be afraid of them. And it also depends on it depends on a lot of things. So it's a little complicated, so you will get some experience. But in general, it shouldn't be a problem. But in some cases, it might be tricky if you are after some new biology, really on the resolution limit. Then it's trickier. Then I would advise is check with super resolution also, or see if anyone else has looked at it with super resolution just to see that it looks okay.
Okay, thank you.
Anyone tried?
Yes, I tried.
Tried?
But I don't know. It's that way. I don't know if my sample is actually what I've seen or it's an artifact. So yeah, I wanted to know.
I think the sample preparation is very important in that case. I don't know what you think, Eric, but people tend to, if they have tissue sections, don't know if you sometimes it looks like you are deconvolving some tissue sections. Sometimes in tissue sections, I mean, most of the time people put them on the slide, and sometimes they put a lot of mounting medium, and that creates some refractive index mismatch, and it means that the point spread function, if you measure, even if you do your job and you measure the point spread function of the microscope with beads or against the cover slip, the point spread function in the sample is going to be different.
Yeah, so in the end, you would have to embed some beads into the sample, really. But it's also that the point spread function is depends on the depth, how deep into the sample. And of course, all samples are different, so it's tricky. But for really complicated samples, I think the confocal is typically better. For thin samples, the wide field can actually surpass confocal after deconvolution. But for thick and complicated sample, I think confocal is typically the winner if you can afford price of the scope and the time it takes to image. But talking about artifacts, I could start with actually I had some extra slides on typical problems, so I could start sharing them before we start the workshop also, if that's okay.
That'd be nice.
No. Okay. I have to give me the permission first.
Sorry. Oh, yes. I always forget that. But, okay, now should be fine.
Right. Go. All right. So so one thing that is always problematic when deconvolving images is what happens close to the edges of the image. And in this case, I'm showing this is a public image from EPF file somewhere where you download the deconvolution lab. So they have this image. So it's shown to the left before and to the right after deconvolution. So there's so yeah, already here you see it's not perfect, and this typically never is. Especially here, you see towards the bottom there's some light pattern and so on. But the interesting thing here is that if you would split this image into smaller parts, you could see what happens close to the edges. So if we crop this image, just take like a quarter of it here, and then deconvolve it, you will see what happens close to the edges. It gets dark. It's dark with the default edge handling settings now in the convolution lab. So that's a problem because it looks dark. There's something there, but it just goes dark. And the even bigger problem is towards the top and the bottom of the image. Because with the default in the convolution lab 2, if you use very many iterations, you go from this raw, and after the convolution, I don't know, this is 100 iterations or something, you will see that all the intensity is gathered in the center. And that's clearly not how it should be. So there are also options in, now is this in the convolution lab 2, to alleviate this. So if you change the boundary handling, you will see this instead. So I think this should be the default here, but it's not. So that's one thing. So many of these artifacts, they can depend on the software that you use also and the settings. And it can be a little tricky to figure out, is it normal in the middle or is it normal to the right before you have some other reference frame also. So unfortunately, it's a little tricky. Maybe with the commercial systems, it's easier, or they avoid these pitfalls. I'm not sure. It's not I haven't used them that much. Right. So I think that's it. Yeah. So we go back here and let's start the workshop instead. So time for you to try out, get your hands dirty, get some experience. And for this means we will do very simple toy images, 2D again. Reason is that 2D images, they are super fast. They will work on your laptops in no time. And it works more or less the same in 3D anyways. So the first task is to get these images from canvas and to convolve them. So the opposite of deconvolution. So you will simulate what happens in the microscope if you put a fluorescent sample with just 2 emitters, and there's a point spread function that looks like this. And let's say, I don't remember how we did last year.
Okay, so please go to Canvas to the workshop and download the 4 images that are there. And then we didn't we have like the GitHub? I can see that the GitHub page is gone, so I will put it again now. So if you open the PDF of the workshop, you will find the GitHub page on the 3rd slide. And this is where you have the workflow, or was it just for the images that you had the GitHub page?
No, it's just for the images, so it's
okay,
the same.
So then you don't need to open that. So you were just showing us how to do it. You were demonstrating how to do it, I think, and then
oh, right.
We followed, yeah?
Yeah.
Okay, so you don't need to go to GitHub, just download the 4 images.
So I think either I can just show how to do this, and then you can get on with the next part.
Yeah, I think that sounds very good. If you show, and then people can follow.
Yep. Right.
So open Fiji.
Yeah, open Fiji. And I even had here a link, but it's the images from Canvas.
So it's the same images. Huh?
Yeah. And for this, you need to have deconvolution lab 2. And also the points, the PSF generator, but I guess it's already been instructed, right?
So all this is already in Fiji, I think, by default, right? Last year it was like this, only I had an issue with it. So if you open Fiji, you will find on the plugins,
I see only the convolution lab 2. The other one I don't see.
Okay, you actually only need the convolution lab 2 now. You need the other one for your assignment, I think.
Okay.
So you can do it later.
Okay.
So this is the thing. If you have image J or Fiji, you just start this plugin. And then either you can just drag and drop the image and the PSF up here. And you choose just convolution noiseless for method. And then you launch. And maybe I even have the images here. So I think...
We can drag and drop the images.
Yeah, you can drag and drop the images.
So
If that doesn't work, it didn't work for me for some reason. Some time I had to go this way. But... So take for image the image called 2_dots, and for PSF the PSF4.tiff.
is everything okay? Everybody okay with that?
Yes. Are we supposed to be in the deconvolution tab because we are supposed to first convolute it?
Yeah. So
Yep.
you open the convolution lab 2, and this is where you will convolute.
Okay.
Yeah. So you go to plugins, and if you can go back a second,
Yep.
Eric.
Yep.
All right. You go to the convolution lab 2 and lab. And then you can drag and drop. For me, it worked to drag and drop the 2-dot images at the top on the first line, the PSF4 on the second line, and then in the drop-down menu, you select this convolution noiseless, and then you click run, and you should get an image.
Well, in the algorithm, I have RIF something, and I cannot really change it. Oh, here I see. I see. What shall I choose? Convolution noiseless.
That's it. And then you
Okay.
click run.
Wait a second. I have the wrong picture. Run. Okay.
Is everybody else okay, or does anyone else have a problem with that?
I don't know what do we need to. So after loading the two images,
Yeah.
what else do we need to do?
Underneath, under algorithm, there is a drop-down menu, and there you select convolution noiseless.
I don't see this drop down menu.
It was difficult for me to find also. You have to kind of open it.
You need to click on the left side of the word algorithm.
Yeah.
Then underneath you see a drop-down menu.
Okay, now and which one?
Convolution noiseless.
Okay, thank you.
And then you press run.
Nice. Thank
Is
you.
everybody okay with that? Looks okay.
Okay. Then we go to the how it should look like. So what you've done now is you took this image, 2 dots, and you could see it as like the super resolution image of a sample with 2 emitters. And then you simulated looking at it through a microscope with the PSF in the middle, so this blob. And what comes out is what you would see in the scope. So you would see these 2 blurry blobs, that's all. So I want you to save this now as something that you remember. For example, 2_dots_x_PSF4.tiff or whatever, just so you remember that this is the convolution of the 2 dots by the PSF4. So now you will have an extra TIFF file among your files.
So it's convoluted.
It's convoluted, yes.
PSF1. Okay.
So this is the ideal scenario for deconvolution, because you know the PSF perfectly, and you have an image which is perfectly noiseless. So it's really ideal. So the next step is, now you will actually do the deconvolution step. So in this case, you will take as input the image you generated, the new image, the blurry one, and you will take the same PSF, and in this case, you will use another algorithm. I suggest you do the Richardson-Lucy with 25 iterations, but you can test whatever you want there, actually. So in principle, you should be able to get back the two dots image or something that looks like this. So let's see if you manage.
You have chosen 25 iterations. How many iterations is a good idea?
It depends. Depends on the size of the PSF, and it depends also if it's 2D or 3D, and on many factors. But here 25 is quite good because I tested it. But in general, it's hard to know upfront, really.
So you have to basically test different iterations and see which one works for your sample test.
Yes. Some software also have automatic methods to stop the deconvolution, typically when there's not that much more that happens.
Okay.
Is there some sort of a rule of thumb for the relationship between the size of the PSF and the number of iterations?
I don't know really. I know in our systems for the 100x microscope, typically 100 iterations are fine with our software. And it's actually like one iteration per magnification.
Okay.
So 40 for 40x, but it's different. It also depends on the purpose afterwards. For example, if you deconvolve nuclei in a DAPI channel, sometimes you just want to be able to segment the contours of the nuclei. And then maybe you don't want that many iterations because you don't want to start to see the nucleoli and other things, because that will be problematic for downstream processing. So it depends on the purpose. And also in some cases, the PSF doesn't match the system very well, and then you can't do that many because then it will start to look strange also. So unfortunately not. But I'm very sure if you do routine deconvolution from the same scope with the same software, after a while you will figure out what works well for you. But in the beginning, at least for me, it was a lot of trial and error also.
Can I ask a question? Based on what parameters should we decide on the algorithm? Here you said like this Richardson-Lucy is good, but how can I make sure that it's also good for my needs?
So the Richardson-Lucy, it's the like vanilla deconvolution algorithm. It doesn't try to do anything fancy, and it's derived from statistical principles to begin with. It should work the best, and you can if you go to the webpage of EPFL, they have a paper where they compare all the different methods in their software, and I think in most cases the Richardson-Lucy is the best one. But then in extreme cases with a lot of noise, or if you know beforehand that there should be no spots, for example, just big structures, there can be other a priori information baked into the algorithm that helps it. But this is unbiased as much as it can be.
Okay, thank you.
and some of them, like the land Weber, it is faster because it doesn't, and it's kind of cheating because it doesn't handle the Poissonian noise properly. It assumes that it's Gaussian noise, things like that. So it's more of a historical interest that they are put there. But I think in those cases, if you use deconvolution lab 2, you should go with the Richardson-Lucy. Okay, but maybe you got something now. Here's what it looked like on my computer. So with 25 iterations, I got back something that looked similar to the first image, 2 dots, but it's a little blurry. All right. So I have a few post exercises for you now. And I mean, just feel free to talk if you have any questions here. But the first is, how many iterations do you actually need before you get back more or less the input, the image you started with, this 2 dots? So as I showed with 25 iterations, it still looks a little blurry. So how many do you really need? So you could try increasing until you're kind of satisfied there. And the other thing is, second, if you deconvolve this image you created at the beginning with a PSF which is smaller, so PSF2, or one that is larger, PSF8, how does that affect the deconvolution? Does it still work? Or what happens? You get smoke out of the fan, your image converted to color, or what happens? So try that. Also, it's an exercise, because then when you're working with real microscopy data, you could try that as well. You use the wrong PSF on purpose, see what happens. That's a way to get familiar with the range of the outputs. And you could do the same also with the PSF8. So I'll just spoil here that if you use PSF8 and convolve it with the 2 dots image, you will not get 2 dots anymore. You will get something that is not really resolved anymore. It will look more like a bar than 2 dots. So in theory, it's not resolved, but what happens if you try to deconvolve it? Can you surpass the resolution limit? And as a last thing, you could also try to create an image to convolve using the with noise option and see how that affects things. So there's no really proposed order here. Just pick anything that looks interesting or do something else. And we will wait a little and I will show you what you would get in all the cases, actually. So I suggest we take like 8 minutes here or something, and you work on your own. And I mean, just pop up any questions you have.
What is the with noise option? Is it the algorithm that we have?
It's an algorithm, yes.
Is it the simulation with noise?
Yeah. Simulation with noise. The second last option.
Yeah, thank you.
And in that case, you have to set Poissonian to 1, I think. The other ones you don't need, but you can try as well. And there should be, maybe there's some. Yeah, so the Poisson option is just if it's on or off, and the other ones is the mean and standard deviation of a Gaussian additive noise in the background. So with real images, you always have this Poissonian, and you will also have Gaussian. but that depends very much on the sensor and how it's cooled and so on, how much of it you have. And on top of that, there are biases specific to CCD cameras and CMOS cameras also, which to some degree can be compensated for before you deconvolve if needed. Like if you have a CCD camera, there can be stripes in the images. For CMOS, there's an individual pixel or a converter for each pixel, so they can have a slightly different transfer function, and in some cases they can completely die, or they can stay on to be so-called hot, always give a high number all the time. And a few of those things can also be more stable, so it will be the same also for the next image. So you would learn actually all the pixels in the camera after a while.
Try the Richardson-Lucy total variation with the default stuff, and it looks like a smiley.
It looks like a smiley.
Yes. With the convolved two dots with the PSF4, and then with the total variation, Richardson-Lucy total variation with default. It's fun. Whereas the other one, the Richardson-Lucy without the total variation is actually really good.
Oh, I have it here.
Did you get the smiley? Yes.
It looks like a smiley. Yeah.
Because yeah.
Something. Yeah, something doesn't really work with that one.
Quite interesting. I'm trying all of them, and it's very different.
Yeah.
I think the Richardson-Lucy is with 10 is it works very well. Just let them know when you're finished, okay? You can just like raise your hand or something so we get to know. going? Does anyone have any difficulty of any sort?
I actually have a question. In the second one, with deconvolute, is the Rick's algorithm, the Rick something algorithm,
So in the
or is
second one, I suggest you do Richardson-Lucy there as well. And
25.
so it's either convolve or convolve with noise, or for deconvolution, just use Richardson-Lucy for this. But if you
Sure,
already have
thank you.
done one of them, yeah, you only need to do one of these, and then we can, I think we need to continue very soon anyways, right?
So maybe we can see what people have done. Maybe next time we can have each group do different things.
Yeah.
Write this down.
Right. So I'll continue, just show what I expected you to get here. So the first question was, what happens if you increase the number of iterations with Richardson-Lucy? And I think you would need more than 500 iterations before it looks like the input. So take some time. So many iterations with this. Now there are other software that doesn't require as many iterations, that do more per iteration. I think, so it's not a general rule of thumb. It's for the vanilla Richardson-Lucy, but it's like this. And the other thing was, what happens if you use this so-called wrong point spread function? So we had this image created with PSF4. What happens if you use the other ones? And with the smaller PSF, it looks like still big blobs. With the larger PSF, it didn't really resolve this thing. This looks like a bar. So it's important. Then this is one of my favorites. If you generate an image with PSF8, you get this thing that looks like a bar. So it's not resolved. The intensity in the middle doesn't drop to 50%, or depending on your way to see resolution. But even that, after 125 iterations, the two dots will resolve, which means that in this synthetic case, deconvolution actually increased the resolution because it was able to separate these dots that were not resolved to begin with. And then there was with noise.
We have a question by Melissa.
Yeah, I actually about the first one. So I was just thinking, like, what is then, like, how do I know it's too much? Because I tried to get it because in the first with the two dots, it was actually just one pixel which was white and all the other one was black. And then even if I tested like 2000 iterations and it was still a few pixels around the white was still like on a gray and not completely black. So how do I then know when I do like what is then too much?
Too much. It is only too much if there's noise involved. If it's noise free, it's supposed to be more or less perfect eventually, and then after some time, nothing will happen. But if there is some Gaussian and Poissonian noise, you will see that it will start to create some other structure that shouldn't be there.
Okay.
But I don't have a straight answer to it. I'm sorry, because I think there's not really a straight answer. Sometimes it's a trade-off. So that was that one, and then lastly is this thing what happens if you add noise into the equation. And with just Poissonian noise, so then the starting point would look like something like this. Even in that case, it managed to work quite well. But if you would add Gaussian noise also, or have a less intense signal then the noise would dominate here, and you wouldn't get any good results, I would say. But I mean, feel free to play around with these toy images or try other real data also. And then this we talked about. And I think the last thing is there's an assignment. Last year it was assignment 8. Maybe that changed.
Yes, it's 8.1 or so, something like this. Let me see.
Right.
Yeah, I just asked.
So test on your data and have fun with that. And if you're, I mean, if you're used to working in the terminal, and especially if you're on Linux, you could also, I mean, feel free to test my program if you want. It's called deconvolve. If you just Google it, you will find a page like this with just some advertisement page you can see
Right.
before or after. And there is some documentation also on this. I think the selling point is that for some things that takes a very long time with the convolution lab, so it's very fast here. Also, it by default it does some choices that make sense also at boundaries of images and so on. So the defaults are quite nice. But it might be a little tricky to get running on Windows and Mac if you're not a programmer.
What are the dependencies?
Dependencies for if you want to use GPU acceleration, you would need OpenCL.
Okay.
Otherwise, all the dependencies are just standard dependencies that you can get with VCPKG in Windows or just with your normal package manager on Linux, and probably also on Mac with Bro.
Is that on Windows and Mac, basically you can't just, it's not like off the shelf. We have to do things after downloading it.
On Windows, there's actually, you can just download it and run it, but you have to do it from the command line. So there's a download link. That's the wrong one. It should go releases here. So there is a file here you can download for Windows.
Oh, okay.
You need to do some other file first, but then it just download and run. But it's only for 3D images, not for 2D. So
Okay.
Okay. Well then. No, I'm on 2D.
Okay. So no, that's
nice.
turned off, and it's another lecture why I've turned it off, because I did that by purpose.
Okay, so you go to your page and then you go, what do? We go get started documentation or release page. Where again was your download page? Maybe you can send it to me.
Yeah, I can send it. It's go to releases on the GitHub.
Yeah. If you can send it to me, then I will put it in the people to test if they want to.
Right. Yeah, sure.
The stuff that they have to do after. Thank you very much.
But otherwise, just use small crops if you have 3D data and you want to use the convolution lab too. It's good. It's fine, but test on smaller crops. Don't do the whole multi-megapixel image directly, because then you would just have to wait a lot. Okay.
Are there any questions for Eric? Get your chance. We have tried, I think during when I was imaging your samples, I think I deconvolved some wide field images for one person. That's all we did so far with deconvolution.
I have a question. Here we had the picture of the PSF
Yes.
as a picture. How do you make a picture of PSF on your own microscope?
So you would use the PSF generator. Maybe I skipped this slice. So there's the other thing from me, EPF. So they have another tool with different PSF models, and you should use here. You should just use the Borna-Wolf 3D optical model, which is the most simple, and it only asks for parameters that you should know about your microscope,
like the NAs at step and stuff like this.
This is in Fiji or this is extra?
It's in Fiji, but you download it also from EPFL. So just Google PSF generator, and you
Okay.
will find it.
Is it by default in Fiji?
I'm not sure if it's by default. Maybe you have to download this plugin.
Yeah, I don't
Actually,
see it
I see it there. So
in Fiji.
at least in that Fiji that I have, that it's there.
But
Yeah.
otherwise, it's just you have to download a jar file and just
Right.
yeah, add it from Fiji. So it's very simple. So you would use the PSF generator. That's the answer, and they will give you something that probably works okay. And if you have thick or complicated samples, or you have an objective that is far from ideal, you might need to do something else or give up deconvolution on that scope.
If I see it correctly, then Born Wolf is for 3D optical model. So if I'm on 2D, I would go to
No, you would use the same. You would just set the size instead to 1,
okay.
assuming that it's still a wide field.
That step I would put to 1. No. What will I put to 1?
No, in the size. Set
Oh,
step doesn't
size,
matter.
XYZ. Now I see. Okay.
Yeah. So just set it to 1 and you will get the 2D image.
I have a question.
Yes, right.
If we have multiple fluorophores, if we are imaging multiple fluorophores, which is the wavelength that we use here to
So
generate the PSF?
yeah, so you should use the short answer is you need 1 point spread function for each emitter. And these models are not super sensitive to the wavelength, so you can probably set it to the emission maxima. But in theory, it can be a little more complicated because there's probably a filter afterwards also that affects the distribution.
Okay, but what do you mean by the maxima, the emission maxima?
The longest wavelength, or is that what you mean? Or do you mean the peak of the
So
Okay.
the peak, I would say.
But you would have to have one for each with each fluorophore, basically.
Yeah.
So
So you would deconvolve each image with its own point spread function. So for each fluorophore, for each channel, you would have the point spread function that corresponds, and then you deconvolve them. Each
Okay.
other.
Okay, thank you.
Did you? I don't know if you did. Did you put any the link in the chat or something? No.
No, I did not.
Possible, then I can.
I could do that, or you could upload this presentation also to Canvas, maybe.
Yes. Isn't it? I think it is already.
Oh, right.
Isn't it? I think.
So I'll just paste it in the chat.
Can I ask a question because it wasn't still clear for me? Are we supposed to write some estimate for exact same parameters of our own image that we want to do deconvolution?
Sorry, I didn't hear what you were saying at the beginning.
So for this PSF generator, is it okay if we do like an estimate, or do we need to like write the exact parameters of our image that we want to do the deconvolution on?
So you will get the best pictures if you pick the correct values, and in most cases you should have that available. But I mean, if you don't, of course you can use it for the assignment, I guess. But if it's for a publication and research, you should get the right values. And also, talking about publications, you should always be prepared to supply the original data also, not just the deconvolved. And so make sure you take notes on what steps you do to deconvolve images so it can be reproduced.
Okay, yeah, that makes sense, of course. Thank you.
Yeah, so I think it is important that, I mean, all the information that are asked, you know about it now because you've gone through the course, and you will be able to find them on the microscope and in the metadata of the images. Which objective you use, what is the numerical aperture, what is the wavelength, etc. So you can input all the correct information, I would say.
Yeah, thank you.
More questions?
Yes, to prepare our images, we need to split the channels, right?
Yeah.
In the case that I have a Z-stack, do I need to do a projection and split the channels, or just simply split the channels?
And if you did, you would just split the channels, and you would get a separate Z-stack for each one of them. So,
and then you can use the 3D deconvolution.
yeah,
Yes.
but it doesn't do multicolor images. So you need to just have a separate image per channel.
Yeah, and those separate images per channel, I save them as TIFF also, right?
Yeah.
Okay, thank you.
I mean, for deconvolution lab, it can also open everything that video image can open. So I think, but for drag and drop, maybe you need to have them as TIFF. I'm not sure. I'm sorry for my video. It just got black here. I'm not sure what happened.
Okay. Are there more questions? Hey, if not, thank you very much, Eric. It was fun.
Yeah, thank you, Sylvie, and thank
And
you, all of you.
thank you. See you again around.